Hello, everyone, and welcome to our North Carolina Botanical Garden Lunchbox talk. I'm Joanna Lalikas. I'm the Director of Education here at the North Carolina Botanical Garden. We're so happy to have you here with us. And today we're really excited to bring you Ben Brewer and Matt Estep from Appalachian State University to talk about Gray's Lily. I'd like to take a moment to thank our Spring 2022 Lunchbox Talk event hosts, James Jocelyn and Beth Hahn, for supporting our Spring Lunchbox Talk series at the Dogwood level. And event hosts for our Lunchbox Talks provide funds that support our program planning, accessibility, and the reach of these talks. So we're very grateful to them. So now I'm ready to introduce our speakers today. Matt will be speaking first briefly. He is an associate professor of biology at Appalachian State University, and his research team focuses on the genetics of rare and endangered plants in Western North Carolina. He's also the preserve steward for the Tater Hill Plant Preserve. And then we'll hear from Ben Brewer, who's a graduate student pursuing his master's degree at App State. He began working with Matt's lab during his junior year, meeting and falling in love with Gray's Lily the following summer. He completed an undergraduate honors thesis collecting demographic data on Gray's Lily, which has grown into his current master's project, and he's seeking a career in conservation of rare plants. Thank you both for joining us today, and now I'll hand it off to Matt to get started.
Good afternoon, folks. I hope everybody is doing well and can hear me okay. My name is Matt Estep. I'm a professor here in the biology department at Appalachian. I'm also the very privileged steward of the Tater Hill Plant Preserve. The property of Tater Hill is owned by the North Carolina Department of Agriculture's Plant Conservation Program. And on the preserve, we have a series of rare plant communities. Those include wetlands like mountain bogs and mountain seeps, as well as high elevation sites like high elevation rock outcrops and grassy balls that are endemic to the Southern Appalachians. Much of my research group's efforts are going into studying rare plants here in Western North Carolina. And to date, 5 master's theses have been conducted on the Tater Hill Plant Preserve in part or in whole. And we hope to work into the future with the Department of Agriculture's Plant Conservation Program to kind of expand the capacity to manage the Tater Hill Preserve and to try and protect those species. Sorry, I have a bunch of things written down. I'm not working very well today. I also have a whole series of undergraduates that are working on the preserve with me as well. A lot of times we're doing biodiversity inventories, trying to get a complete understanding of the flora of Tater Hill. We set up plots to look at pest management, and we also are beginning this year to do pollinator surveys and studies where we're trying to understand the pollinators and plant visitors that are on the preserve to better understand how gene flow might be working in that way. I hope that today's discussion is exciting for you. The idea was to let you hear directly from the researcher about some of the work that Ben is conducting on the preserve, and then have you guys come up later in the summer when the plants are blooming so that you can actually see these plants and interact with them in a more personal way. I know that the field trip has already filled to capacity. We do try and keep those numbers on the small side to protect the preserve. You can reach out to me personally via my university email, and I can try and set up some sort of an independent tour later in the summer. I'm not sure we'll be able to hit the Gray's Lily, but there'll be other things to see, of course, always on the preserve. And yeah, so without much further ado, let me introduce Ben briefly. Again, Joanne already spoke of some of his accomplishments. Ben and I started working together in about 2017. He very quickly proved himself to be an excellent researcher, and he developed his own honors thesis where he began looking at the demography work within Gray's Lily. He graduated in 2020, well, I guess 2019, but he was awarded the Wayne Van Deventer Award through the biology department here. This is our highest award that we can offer to the ecology evolution students for having the highest GPA of that particular year. Ben also published his first manuscript that year, where he took an independent study and his GIS certificate and was able to turn that into a publishable product that we're very proud of him for. Ben is scheduled to complete his master's research in or defend his thesis before December of this year. And we're very excited to hear about some of the work that he's been conducting. I also want to put a pitch out there that he is looking for a doctoral program. And if there are any people on the call today that have a position available, I highly recommend Ben. So without further ado, Ben, please let us know more about conserving grazed lily through genetic diversity analysis and demography analysis.
All right, thank you for a wonderful introduction. So hopefully we're seeing a pretty slide, and now hopefully we're seeing a fancy little laser. So yeah, hi everyone, I'm Ben Brewer. First, just want to thank the Botanical Gardens for letting us have this really great opportunity to talk about some of our research work, and to introduce the real star of the show, which is grazed lily, or Lilium grayi, one of our most stunning Southern Appalachian endemic species. It has been an absolute pleasure and a privilege to work with it the past few years, and I'm excited to talk to you and how I've tried to protect it in as many ways as possible. So up in Boone, we hear the term Southern Appalachian endemic a lot because we do live in one of the hotspots of biodiversity in our country. Our region is home to over 10,000 species, many of which are endemic, meaning occurring here and nowhere else in the world, primarily due to a complex history of glaciation over the past 30,000 or so years. So during the last glacial maximum, as glaciers advanced southwards from the poles and ended up covering down into New York and northern Pennsylvania, northern species that lived in New England and Canada were forced to migrate southwards and advance the glaciers. Then when the climate rewarmed and those glaciers retreated back up towards the poles, many populations of species retreated northwards, following them and reestablishing their original ranges, but many populations retreated up into the mountains, seeking out those cooler climates at higher elevations. And so this, combined with the fact that our region was never completely glaciated or never covered in glaciers, has created a region that is rich with species, but also one that is inherently at risk to future changes in climate. And that's what this map is showing us. Produced by Jenkins et al. In 2015. They produced what they call a priority score, which is a function of how many endemic species are found in an area and how protected or conserved their remaining habitat is. Those areas in orange and yellow and red are those with a high priority score. And as I think you can see, our own Southern Appalachians were determined to be the #1 priority for further land conservation because of our threatened endemic species. And so I do want to back up for a second to talk about why this biodiversity matters, because one of the more common questions I get that I honestly don't love hearing is something along the lines of, okay, well, why does grazed lily matter? Why should we bother saving it? What's the point of all of this? And, my knee-jerk response in my head is kind of something like, well, I just think all biodiversity is worth conserving for its own sake, but there is more to it than that. I think we all, or at least I hope, we are all aware of the climate crisis and global warming facing our planet. But many people don't realize we're also facing an equally catastrophic biodiversity loss crisis as species rapidly go extinct. There's clear evidence that conserving biodiversity is critical to maintaining the ecosystem functions and services that power our planet. And that's what this graph is showing, produced by Cardinale et al. In 2012. It's an article from Nature, Biodiversity Loss and Its Impact on Humanity. I read this paper a few years back, and this graph, I think about it nearly every day now. We see on the right side of the graph, with high biological diversity, we have high ecosystem function. And then as we lose biological diversity, ecosystem function drops as well. And I hope you can see the slope of that line is getting steeper. So the loss of ecosystem function accelerates. It gets more and more rapid as we lose more biodiversity. This is because biodiverse communities of plants and animals are likely to contain many different species that utilize different partitions or different kind of segments of natural resources. And that increases the overall efficiency of nutrient uptake in the system. We know that increased species richness or number of species in primary producers, those things that conduct photosynthesis, leads to higher efficiency of nutrient uptake, all up the food chain in both terrestrial and aquatic ecosystems. Additionally, having biodiversity in our ecosystems increases their stability or resilience over time as natural disturbances, environmental stochasticity, random events like hurricanes, floods, wildfires, tornadoes, all the things that we don't love, they are unlikely to affect all species equally. So by having a lot of different kinds of species in an ecosystem, there's a lot of that redundancy built in. And so it's kind of similar to the concept of risk spreading or just not putting all your eggs in one basket. Altogether, the continued functioning of life on Earth as we know it is inextricably tied to its biodiversity, and thus conserving that diversity must be among our greatest priorities. A beloved quote for conservationists is from Aldo Leopold, one of the founders of the Wilderness Society, author of a Sand County Almanac. And he said, "To keep every cog in wheel is the first precaution of intelligent tinkering." Meaning, we're still learning how our ecosystems function. And if things go extinct before we have a chance to understand how they work, we're never going to. And that gets us into Gray's lily, which I think is emblematic as a Southern Appalachian endemic species. It was discovered by one of our most esteemed botanists, Asa Gray, in 1840 on Roan Mountain, and he determined it to be Lilium canadense, the Canada lily. It wasn't actually until a few decades later that his student at Harvard, Serena Watson, correctly, in my opinion, described it as a species, Lilium grayi, based on its horizontal to nodding flowers, as opposed to the pendant flowers of the Canada lily that point straight down at the ground. And he named it for his teacher, Asa Gray. Contemporaries did doubt that it was its own unique species, and many people considered it to just be a brood of the Canada lily that had smaller flowers and flowered earlier in the year. But history has sided with Serena Watson, and it's considered a unique species to this day. However, there is a red flowered form of the Canada lily, Lilium canadense variety editorum, and there's just been perpetual confusion between those two species to this day. As recently as the early 80s, Gray's lily was listed as existing in Maryland and West Virginia due to incorrectly identified populations of the red flowered Canada lily. Furthermore, in southwestern Virginia, where Gray's lily and the Canada lily grow sympatrically or grow side by side, they're suspected of being introgression or hybridization between the two species, which is further complicating the species boundary between the two. Thinking about Gray's lily kind of in just a purely botanical sense, it's a member of the Liliaceae family. It's a fairly long-lived perennial species, taking between 3 to 7 years to become reproductively active. The species overwinters as a scaly bulb underground. It emerges in the mid-spring with vegetative tissue, produces these gorgeous flowers in June and July, and then it produces its fruits, which are capsules, in August through October, before the above-ground tissues senesce or die back to for the year, kind of like trees dropping their leaves in late fall. Just a small kind of botany terminology note. Gray's lily has tepals, which is the term for when the petals and sepals of a flower look alike. So, when we look at these flowers, it might appear like they have 6 petals, but it's actually 3 petals and 3 sepals. And all this to say, from now on, when you hear me talk about tepals, just remember that I'm talking about the petals. So the flowers are this gorgeous kind of red to deep orange color. They have splashes of yellow and orange on the inside, as well as those gorgeous purple to reddish brown spots. The flowers are fairly small, between 3 to 5.5 centimeters long, or roughly 1 to 2 inches long. The tepals, remember the petals, are slightly reflexed, meaning they are at the tip of them bent back slightly towards the base of the flower. And this is one of the distinguishing characters of Gray's lily. If you think about ornamental lilies, things like the Easter lily or the tiger lily, they have strongly reflexed tepals. The tepals are bent back, nearly touching the base of the flower. That is not the case for Gray's lily. The leaves grow as whorls, meaning that 3 or more leaves come out of the stem at each node or each place where leaves come out, kind of in a circular pattern. Their fruit is a capsule. It is a trilocular dehiscent capsule. Trilocular meaning there are 3 little compartments inside that contain seeds, and dehiscent meaning at maturity it splits open on its own, as we can see in these photos, kind of at different parts of the process on the right. Each capsule contains an average of 200 seeds, which are flat and winged, so perfect for being distributed by a strong breeze. The only reliable pollinator known for the species is the ruby-throated hummingbird. They typically like red flowers, and Gray's lily is perfect for them. The species mostly grows in mesic or wet habitats, including high elevation bogs, seepages, moist forests, wet meadows, but it also thrives and is really known for growing on the more xeric or dry grassy bald plant community. It's restricted to high elevations. The highest elevation population occurs at Grassy Ridge Bald on Roan Mountain at 6,184 feet. There are a handful of populations around 2,500 feet, but those are all in southwestern Virginia and are those populations that are suspected of having hybridized individuals and some Canada lily individuals. So a bit of an open question about how much we should consider those true Gray's lily. The lowest population that most people would agree are 100% pure Gray's lily occurs at 3,500 feet. Gray's lily faces many of the threats typically associated with endangered and threatened species, including a limited geographic distribution and range, continued habitat loss and poaching and other anthropogenic effects, loss of genetic diversity due to genetic drift in small and isolated populations. But likely the greatest danger to the species comes from a more specialized enemy, and that is lily leaf spot disease. So lily leaf spot disease results in plants wilting and prematurely senescing, or dying back to those below ground tissues earlier in the year than they would normally. This happens on such a wide scale in populations that it causes an event or a phenomenon known as the early season population collapse. And that's what we see in this photo here. Hopefully we can see that the vast majority of these plants are yellow and brown, and even a non-botanist would know that's not really healthy. And this photo was taken in mid-June. So, plants should be actively flowering. It's still months away from when they would produce fruit, and many of them are already dying back for the year. Lily leaf spot disease was first studied by Moni Bates of the North Carolina Plant Conservation Program at Bluff Mountain and Sparta Bog in North Carolina in the late 1990s. And she found that the disease caused reduced reproduction due to the premature senescence. Then in 2011 and 2012, Russell Ingram of Eastern Tennessee State University, conducting demographic monitoring on plants along a transect at Roan Mountain, again, finding the majority of plants were prematurely senescing to the disease and not reproducing. And he was able to determine that the invasive ascomycete fungus, Pseudocercus virella inconspicua, was the pathogen responsible for the disease. So Pseudocercus virella inconspicua, or lily leaf spot disease, which is a lot easier to say and what I will call it from now on, is a Lilium-specific pathogen, meaning it only affects members of the genus Lilium. It was originally described in 1884 on the Eurasian species Lilium martagon. The disease appears first with these tan or yellow lesions on the leaves. These are caused by fungal canidia, which we can see here magnified 90 times on a Grays lily leaf. As the disease progresses, those lesions radiate throughout all above ground tissues, eventually causing the plant to, again, prematurely senesce most of the time before it's able to successfully reproduce. And so we kind of end up with this morbid race between the plant trying to successfully produce capsules and reproduce, and the disease working its way up to try and take out the plant, essentially. And unfortunately, the disease wins a lot of the time. Russell Ingram and another student from that lab, Cindy Barrett, conducted an extensive search of herbarium records to try and determine the first time that we could see lily leaf spot disease here in North America. And they found it on a Canada lily individual from New England in 1916, with the first occurrence on Grays lily on an individual from Roan Mountain in 1947. Following that, Cindy Barrett, her master's thesis was able to confirm that lily leaf spot disease is present across the range of the species. So we know that it is everywhere. Another threat facing the species, I've mentioned a couple of times that there's this likely hybridization going on with the Canada lily producing hybrid pseudo-grayi individuals. And because the Canada lily is much more common, especially up north where the two species grow sympatrically or side by side, there's a suggestion that genetic swamping or the erosion of alleles that make Grays lily what it is are going to be a long-term threat to the species. The hybridization occurs as a cline or a transition zone along the Blue Ridge Parkway, with Grays lily slowly integrating into Canada lily with increasing latitude from south to north. So on the left, we have what we think of as a pure Grays lily individual from Roan Mountain. We see that it has those nodding flowers that Serena Watson originally used to distinguish the plant. It has those slightly reflexed tepals to the point that the anthers are completely contained within the bell shape of the flower when you look at it from profile or from the side. On the right, we have a Canada lily individual from the northern end of the hybrid zone. We see that it has pendant flowers that are pointing straight down at the ground rather than being horizontal or nodding. Its tepals are much more strongly reflexed to the point that the anthers are actually exserted, where they're poking out of the bell shape of the flower. If you remember, I mentioned the ornamental lilies like tiger lilies. This is more akin to that, where those tepals are bent far back. Then in the middle, we have two plants believed to be hybrids from different places in that hybrid zone. On the top, we have an individual from the southern, more Grays lily end of the hybrid zone from just across the North Carolina border. We see that it has those slightly reflexed tepals, anthers that are nearly contained within the bell shape of the flower, but the flower is pendant, pointing down at the ground, and is also much larger than we would expect for a Grays lily plant. I know that's hard to see here. And then on the bottom is a likely hybrid from the northern or the Canada lily side of that hybrid zone. And I think we can all see, it looks a lot like a Canada lily flower. It has pendant flowers, strongly reflexed tepals, exserted anthers, but the size of the height of this plant, as well as the size of the flowers, was very small, pointing more towards Grays lily, much smaller than we would expect for a Canada lily individual. So all in all, more kind of investigation, more work is needed to really identify what these plants are in these likely hybrid populations. Looking at the geographic distribution of Grays lily, we can see that it is clearly a Southern Appalachian endemic species occurring only in the mountains of Virginia, Tennessee, and mainly North Carolina. These 17 counties in green are those that contain either a current or historical population of Grays lily. However, the current distribution is smaller than that. NatureServe now lists there being 46 extant or still surviving populations of Grays lily spread among 15 counties. And I believe this is still an overestimate, as I visited 4 listed Grays lily populations that had no flowering individuals. One of the things that really surprised me when I gained access to the data and records of graze lily populations is how many of them hadn't been visited in at least a decade and some in multiple decades. And so I think it's likely that of those 46 occurrences, a number of them have just winked out over time, either succumbed to the disease or something else. So the true number is lower than is listed because we need more data on these populations. Graze lily is listed as a conservation concern by pretty much every relevant agency. NatureServe lists it as critically imperiled. It is a federal species of concern and is listed as threatened, endangered, and imperiled in North Carolina, Tennessee, and Virginia, respectively, which again are the only 3 states where it occurs. One thing that I think is really important to keep in mind when looking at a county map like this is that it can be kind of misleading because when you see an entire county lit up in green, it might seem like the plant occurs everywhere there, when it's very possible that it's just a single population of 10 to 15 individuals in a county, and that's it. So take these county maps with a grain of salt. So these circles and diamonds are the actual general locations of the sites where I did my demographic monitoring work for my research. So my primary objective for my work is to conduct a range-wide health assessment for as many graze lily populations as possible in 2 ways. First, a genetic diversity analysis using microsatellite markers or DNA fingerprinting to obtain genetic diversity health metrics that will tell us, which populations have a lot of diversity and which don't, as well as investigate population structure or interconnectedness between populations. What is more similar to other populations? Where do we think the gene flow is going? Things like that. Additionally, I've conducted demographic monitoring of populations, conducted over 2 visits per season to try and get an idea of how many flowering plants are successfully reproducing and some of the effects of lily leaf spot disease across the range of the species. So over the past few years, I have visited a total of 29 listed graze lily populations of those 46 total. I successfully found flowering individuals at 25 of them, and I conducted demographic monitoring at 22 of them. This does include 6 populations thought to be experiencing that hybridization from the Canada lily in southwestern Virginia, represented here with the diamonds. And I'm also hoping to use my collected morphological and molecular data to try and do a little bit more investigation of that hybridization issue between the species. Additionally, I have kind of picked up some fun kind of bonus conservation side projects along the way that I'll be talking about. So my demographic monitoring protocol, again, entailed 2 visits to populations per season. First in the early summer when plants were producing flowers, and then revisiting a couple months later during fruit or capsule production. On my first visit to populations, I collected general plant health data, including the height of the flower in centimeters, how many leaf whorls each plant had, how many of those leaf whorls showed signs of lily leaf spot disease, how many flowers each plant had, and then some data on the surrounding vegetative cover from the ground up to 1 meter, from 1 meter to 2 meters, and then canopy cover. And this vegetative data was broken down into 10 classes based on the Carolina Vegetative Survey plot system. On those first visits, I tagged each plant with a small aluminum tag for identification, took a GPS location, and took a small leaf tissue sample for the genetic diversity analysis. And then returned to populations a couple of months later to rediscover my plants, determine the number of successfully produced capsules, as well as the overall state of the plant at the end of the season. Importantly, lily leaf spot disease is something that is transmissible by humans, both by our touch and by our clothing. So all clothing that I wore was washed in between visits to populations, and all measuring equipment and footwear was sterilized with 90% ethanol between visits to populations to try and prevent further spread of the disease. I mentioned that there were some extra fun things I did along the way. I received permission to collect seeds from mature plants at select populations for conservation at the North Carolina Botanical Gardens Seed Bank. And additionally, our monitoring at the Tater Hill Plant Preserve was conducted in 8 plots that were 25 square meters or 5 meter by 5 meters. They were set up in pairs. So 4 plots were located on the grassy ridge bald at the highest elevation on the property. And then 4 plots were set up under a hawthorn forest at slightly lower elevation. 1 plot in each of those environments, so 2 plots total, received a 6-foot tall deer fence or deer exclosure around them to try and start figuring out the effects of herbivory and browsing by deer on the species. Additionally, we set up a trail camera pointed at each plot that was programmed to take a burst of 5 photographs whenever motion was detected to try and get a better idea of what was going on in those populations. So these are the results from my first visits to populations for each year. The project technically started in 2019 as an undergraduate thesis on a much smaller scale, and pretty much just made me realize that I need to go to graduate school and spend more time working with Gray's Lilly. So when we look at the larger sample sizes from 2020 and 2021, we see that the average height of flowers was just below 90 centimeters. On average, plants had roughly 6 whorls per plant, of which a quarter, or 24 to 26%, showed signs of lily leaf spot disease. The majority of plants had a single flower. However, there were some highly productive individuals that did push the average a little bit higher. And importantly, lily leaf spot disease was detected or was seen at all sites visited in all years visited. So I was able to verify Cindy Barrett's work that it is present across the range of the species. This is worrisome on its own, but things start to get a little more dire when we look at the results of my second visits to populations. And here we see the majority of plants did not successfully reproduce in any year of monitoring, with slightly less than 1/3 of plants reproducing in 2020, and only 13% of plants producing capsules in 2021. This was primarily due to premature senescence from lily leaf spot disease. However, herbivory or browsing, likely from deer, also played a pretty serious part as well. Other no capsule plants just refers to plants that were mowed because they were on the roadside, or plants that just appear to have not been pollinated. Only plants that I was able to successfully rediscover were used in these calculations. I was able to rediscover about 93% of my tagged plants, though I should say, I do suspect that many of those non-rediscovered plants did prematurely senesce to the disease. In 2020, there were 2 populations that had absolutely no capsule production. And in 2021, there were 7 populations with no capsule production. And so obviously this is all alarming, but unfortunately it does still get a little bit worse when we remember that 6 of my populations are those thought to be hybrids or to contain hybrids and some Canada lily individuals. And so when we take those plants from those populations and separate them out, we see the results change a little bit. And so that is what these graphs are showing. On the left of both graphs, these gray bars show the combined total of all plants. So those are the same numbers that we saw on the previous table. The green bars are those plants from populations that we think of as being pure Gray's lily. And the orange bars are from plants that we think of as those pseudo-grayi likely hybrid populations. And so when we do this, we see successful reproduction plummet from 32% to 22% in 2020. And from 13% to just over 8% in the pure gray populations in 2021. Unsurprisingly, this is due to an increase in premature senescence from lily leaf spot disease, as we see over half of those pure gray plants prematurely senesced in 2020, and a whopping 86, nearly 87% of plants in 2021. The fact that such a smaller proportion of those likely hybridized plants are prematurely senescing to the disease resulted in a higher percentage of them, a majority of those likely hybrid plants, successfully reproducing in both years. And this may seem good at first glance, but it's actually worrisome because the fact that those likely hybrids are reproducing at such a higher rate relative to the pure gray eye plants again suggests that genetic swamping or the erosion of Gray's lily alleles are going to be a long-term threat to the species, certainly in those likely hybrid populations. And so, since a lot of this analysis hinges on those differences between the two groups of plants, our next question is, can we detect statistically significant differences between the two groups? And so to investigate this, I ran a series of Welch's tests, which are essentially a two-sample t-test that is much more conservative in its predictions because of the fairly large difference in sample sizes between the two groups in my data. I know not everyone here is a statistician, so the important things to look at are the means or the average for each group, as well as the p-value of the test. If a p-value is greater than 0.05, like this one, we say that the test is non-significant and that the differences between the two could be because of random chance. If the p-value were less than 0.05, we would say that's a significant difference that is not just due to sampling error, random chance. And so we see that there's not a significant difference in the number of flowers produced, with 1.52 flowers per plant on average in gray eye to the 1.67 on average in those likely pseudo-gray eye populations. Where we do start to see significant differences is in the height of the plant, with the pseudo-gray eye being significantly taller at 100.6 centimeters to the 85.8 of pure gray eye. Difference in the number of leaf whorls per plant, with 6.9 per plant to the 5.6 on average of gray eye, and somewhat expected, but most interestingly, a significant difference in the number of fruit or capsules produced, with the pseudo-gray eye plants producing 0.737 capsules per plant on average, and the pure gray eye only producing 0.260. And the fact that we see the significant difference in fruit produced, but not in the flowers produced is a big deal, right? Because both groups are producing the same number of flowers, but those likely hybrids are reproducing at triple the rate of the pure gray eye population, which again is pointing to that danger of genetic swamping issue. And so all of this really adds to the body of evidence that these populations that we think of as hybrid are in fact hybrid, but what we really need is that molecular DNA data to finally elucidate what's going on in these populations, and that is forthcoming. So moving on past the demography work, I was lucky enough to receive permission to collect seeds from plants for conservation at the wonderful North Carolina Botanical Garden seed bank. I was able to collect a total of 690 seeds from 10 different plants, all located on the Roan Mountain Massif. I collected leaf tissue samples for the genetic diversity analysis from all of these plants. So we're going to have that maternal genotype data, which is pretty exciting as well. Especially exciting to me is the fact that I was able to collect seeds from these first three plants in blue, 10439, 10440, and 10441. These were three of my favorite plants from all of my work, and so I would like to introduce you to them. Here on the left, we have 10441. On the right, we have 10440, and there in the background, we can see 10439. And these were three of my favorite plants for a couple of reasons. First, I think you can see they're right next to the road. So I hiked about 150 miles in the first half of my field season, both years. And these three plants, or these two plants in particular, were the most spectacular plants I saw anywhere across the entire range of the species, right next to the road, like a mile down from Carver's Gap, which just cracks me up. But the real reason that I love these plants so much is that together they produced 26 flowers, all of which successfully matured into capsules and produced seeds. These 26 capsules, in 2020, across my 15 sites, I saw a total of 128 capsules. So these three plants with their 26 represented 20% of all of the successful reproduction that I saw that year across the entire range of the species, right here, right next to the road. So incredibly important plants. I visited them 7 or 8 times in September and October that year, waiting to collect those seeds. I consider them dear friends. I love these plants, and I was very excited to collect nearly 350 seeds from them for that ex situ conservation. Thinking about those deer fences, exclosures that we built at Tater Hill Plant Preserve, unfortunately, due to the plots have so few plants in them that we don't have enough data to run any statistically robust tests yet to say strongly if they're working. But the preliminary data is definitely suggesting that it's good news. In the 2 seasons since we constructed them in February 2020, the only plants from my monitoring to successfully produce capsules were located in this plot on the right, inside the deer fence on the grassy bald. So obviously, again, preliminary data, but suggesting that the fencing is helping. And then looking through the photos from the trail cameras, we get some fun insights on how things are faring up there. One of the most exciting was that we saw this hummingbird, and hummingbirds fly too fast for the trail camera, so that's what this weird thing is. But we saw the ruby-throated hummingbird flying around the grassy bald plot with a grazed lily in the corner. And in the same burst of photos, we caught it actually visiting the flower and pollinating, which is really exciting. You love to see your plant being pollinated by its pollinator. The main thing, is that we built it for the deer, or rather to keep the deer out. And so we saw plenty of deer. We saw does, we saw sweet little fawns chasing after mamas, we saw bucks. But the most important thing is that in our hundreds of deer photos from the 2 years, we don't have a single photo of a deer inside the fence. So we do feel strongly that the fences are effective in keeping the deer out. And again, that preliminary data suggests that it is helpful for the reproduction of the species. So we've talked about a lot of results so far. Kind of our key takeaways to this point are that lily leaf spot is present at all sites visited, in all years visited, and in those populations that we think of as pure gray eye, the majority of plants did prematurely senesce to the disease. Overall, capsule production is very low. Again, it is significantly lower in the populations that we think of as pure gray eye. And the fact that we see that higher capsule production in those likely hybrid populations suggests that genetic swamping is going to be a long-term threat. Because a small proportion of flowering individuals successfully reproduce, sites that appear healthy or robust with a lot of flowering individuals may be misleading, since so few of them are actually producing seeds. And then the other side of this is that those small and isolated populations are clearly in serious trouble. As there were 2 populations that produced no capsules in 2020, growing by 5 for a total of 7 populations that produced no capsules in 2021, including those same 2 from 2020. So they produced no capsules 2 years in a row. Moving on to some happier news, I was able to collect 690 seeds from those 10 plants for ex situ conservation, and the preliminary data from the deer exclosures suggests that they are helping reproduction for the species at those populations. So if I've done my job at all, hopefully everyone is sitting there thinking like, oh God, grazed lily is in trouble. What can we do to help? And the obvious answer is go to grad school and get a master's degree working with grazed lily, right? Yeah, because we can all do that. No, obviously not. But the good thing is there are things that we can all do. And the big ones are to stay on the trail when you're out hiking, not to touch or pick wildflowers, and maybe most importantly, to educate everyone you can about these things. I think the value of education out on the trails and in our natural areas, it's invaluable. It's truly priceless. The sign on the left is from the Blue Ridge Parkway, reminding people to stay on trail when they hike. When trails are built in sustainable ways, we hope that they avoid populations of rare plants. And so when people go off trail, they undo that sustainability work, and they're likely tromping through populations of rare plants that, if they're not flowering, most people have no idea what they are. And the other big thing that is true for Gray's lily in particular, but also likely true for many rare species, is to not pick or touch the flowers. Appreciate them with your eyes, not your hands. And the big reason for that is that we know that lily leaf spot disease is transmissible by our touch and by our clothing. And so the Southern Appalachian Highlands Conservancy, the U.S. Forest Service, and the Appalachian Trail Conservancy put up these amazing signs all along the Appalachian Trail at Roan Mountain, and they work. When I'm out there working, you saw me, I have my big hat, my backpack, a camera, and a GPS unit. I look like a big dweeb. And so people are very curious about what I'm doing. And when I tell them that I'm working with Grace Lilly, almost all of them say, oh, it has the disease. Don't touch or pick the flowers. They had read the signs, they had some idea of what was going on, and they knew how they could help, or, more importantly, how they could not hurt. And so, again, just the value of this signage cannot be overstated. It's a big deal. People out in our natural areas, they're there because they love nature. They don't want to do damage. We just don't always realize how much damage that we can do. So the other side of that is something like this. As we know, Gray's lily has those nodding flowers, and they're absolutely gorgeous on the inside of the flower. And so people's natural inclination when they want a picture is to grab it at the base and turn it up so they can snap a photo. And by doing so, if they have lily leaf spot on their hands, and there's no way to know if you do, if you just graze a leaf, you probably have it on there. But by doing this, they provide the plant or the disease a shortcut up the plant, and basically makes it very likely that it's going to prematurely senesce for the year. And so this observation, they were kind enough to tell us it was near Callaway Peak. And so as soon as I saw this is individual number 10901 at Grandfather Mountain. I had been here about a week prior. I know exactly where this plant is. I know this plant. And as soon as I saw this observation, I realized that it was likely going to be senesced for the year and not produce fruit. And when I returned in September, of course, it had prematurely senesced and did produce fruit. So please don't do this. This is a no-no. Again, appreciate the flowers with your eyes, not your hands. And then, revisiting these three special plants that I love so much, these plants that I love so dearly that are clearly important to the overall health of the species. I was so excited in 2021 to revisit them and see how they were going to do this year. I visited Roan Mountain early in the season on June 13th, kind of like a scouting run. And I walked down to find them, and here they are circled in red, these just beautiful plants that were bursting with buds. One of them had 15 buds, another had 13, and the third one had 12. So it promised to be an even bigger year in 2020 than 2021. Throughout my field season, I saw multiple iNaturalist observations that I knew were these plants. I know the background, I know what they look like, and they look like they were thriving. And then on July 13th, I went up to do my monitoring at Roan Mountain, and I walked down to see my plants, and they were gone. I could see where there were tire tracks right on the side of the road, and I could see heavily disturbed vegetation right around where they should be. And it is pretty apparent to me that somebody drove up and dug them up and that they were poached. And I say that because, a lot of times when I talk about plant poaching, I can tell people don't take me seriously. They kind of roll their eyes and they think, oh, okay, yeah, people are going to drive out and just dig up plants. And yes, they absolutely do. And so, the North Carolina Native Plant Society has an amazing website where they list native plant nurseries all around the state that are known to, practice sustainable practices. And so if you want wildflowers, please go to them to buy wildflowers. Don't dig them up off the landscape because in a situation like this, these plants were incredibly important. They produce so many capsules, and the fact that they are no longer on the landscape is just heartbreaking. So lastly, to think about is the future directions for this project. And that is the genetic diversity analysis. Over the course of the 2 years, I collected tissue samples from 618 plants. I've just recently completed my DNA extractions on all of them, which is a massive and amazing milestone to hit. And now I'm moving into the DNA fingerprinting with microsatellite markers to do the rest of the genetic diversity analysis. And again, this will give us metrics on the different genetic health populations and to help us understand what pollinations are kind of receiving cross-pollination, how does gene flow occur. There's a lot of interesting things that we're going to find out that is forthcoming. And with that, unfortunately, I'm butting up on time, so I can't thank all of the wonderful people who have helped in this process. I know many of you are here in the chat, and it's wonderful to see you. The conservation community in North Carolina and Virginia, just full of incredible people, and I received great help along the way. But I do want to make sure I thank the North Carolina Native Plant Society for funding through the Tom and Bruce Shin Grant, the Appalachian State Office of Student Research for funding through for funding the project, everyone in the Dr. Estep's lab and Dr. Estep himself for being an incredible role model, and the Nobles Family Fund for funding the genetic diversity work and for just funding so much incredible work that we do in our lab. And with that, I would love to take some questions and talk about Gray's Lily some more. Thank you.
Awesome. Thank you so much, Ben. When I first saw this title and description, I was so scared that this information would go over my head. So thank you for making this accessible to those of us who don't have as much knowledge as you do, you and Dr. Estep's lab. We have a lot of questions coming in. I'm not sure that we'll have time to get through all of them, but I'll ask a few that stand out. One, you mentioned hybridization as an issue for Gray's Lily. Are you also monitoring inbreeding as an issue for future conservation, considering the overall small population size?
Yeah, great question. And that gets right into the heart of the genetic diversity analysis. That is there's something called an inbreeding coefficient or an FIS statistic. And that is something that we'll be able to calculate, one of those diversity metrics I talked about, once I finish the genetic diversity analysis, because that's a huge question, right? We know that in plants, inbreeding is a bad thing, just like in humans. And those small and isolated populations are probably going to have a lot more inbreeding there. And so that will kind of give us ideas of, if there's a high amount of inbreeding there, that's going to be what we consider a less healthy population. And then, augmentation is a big, scary word. We don't always like to move plants around the landscape, but, if it were to ever be necessary, that genetic diversity analysis work, if we find populations with low inbreeding, those might be good candidates for source populations to, take some seeds or take some plants and move them to the genetically appropriate populations. So yeah, inbreeding is a huge deal. Great question. Something that I will hopefully know about for sure in a couple of months.
Wonderful. You talked about the future of this project, and one of our attendees also has a question about that. Are your monitored plants marked precisely enough for long-term demographic monitoring? Are there any plans for that, and could it potentially be a citizen science project?
Oh God, I'd love that. At Tater Hill Plant Preserve, yes, all of the plants are marked with staples in the ground for long-term monitoring past my research work, and I know Matt is very excited to continue that monitoring as well. That was the only place that I put down permanent markers. Everything else was temporary, so unfortunately nowhere else. But yeah, I mean, this project was originally designed in 2018 and 2019 when I came into it. It was designed to be a community monitoring protocol, and I can find the data on that. But the idea was we give out the instructions on how to do the monitoring in data sheets to all the different places where there are Gray's Lily populations. Everybody monitors their own site, and then we compile all the data together to do this work. And unfortunately, we didn't have as much buy-in as we wanted. And so that's why I kind of made it my master's to go out and do it all myself. Well, not myself, do it with majority of me with a lot of great help. But all that to say, yes, it would be a great citizen science project. We have that kind of sheet explaining how to do the demographic monitoring and then data sheets. I'm going to put my email in the chat. Please, if you're interested in doing that, shoot me an email and I can get that data to you. brewersb@appstate.edu. I should probably have that on a slide somewhere. But yeah, please feel free to email me asking about Gray's Lily, anything, and I'll try to get that information out. Good citizen science project.
Sounds great. And we'll share your email address in the follow-up email that goes out to participants as well.
Good.
I think we have time for one more question. You spoke, a ton about hybridization, but could you speak on the difference between hybridization and evolution?
Oh boy, that's a big question. And it depends on who you ask, right? There's yeah, I mean, I would say if biodiversity wasn't being lost at such a rapid rate right now, then it would be interesting to sit back and look at this and see, would in the native kind of situation, would Candida lily kind of slowly, I guess, cannibalize grazed lily and then lead to further divergence from each other over time? We know certainly, in polyploid species, we would call it recurrent polyploidy, but we know that that's kind of a function of evolution in plants, that things hybridize, become just this kind of hybrid species, and then over more geologic time or evolutionary time, split back out again. And that is a natural process of evolution. Again, my kind of concern is that due to anthropogenic impacts, things are going extinct at a rate that we've never seen before on our planet. Well, I guess excluding, the big mass extinction events, which we're now in one, but we're causing things to go extinct at such a fast rate that, I think it's still worth saving grazed lily. I know that's not the kind of crux of the question, but gosh, yeah, I mean, I can talk for 15 minutes on that question. That's a really good question. They are certainly similar aspects. My fear is just, I want to keep all the species that we can. Yeah, I feel like I'm not answering this very well because it's too good of a question and I need more time to answer.
We appreciate your words.
All right, well, thank you so much, Ben, and also Matt, for being here and for sharing all of this information about the grazed lily and for all that you both are doing around conservation of this plant. Thank you. And we're grateful to have you join us.
Yeah, like I began with, just having this opportunity is wonderful. Thank you guys for all the great work you do. The seed bank here or at the Botanical Gardens is a big part of my research work. So thank you for the opportunity and everything you do.
Great. Well, to our audience, thank you all for joining us today. If you're not currently a member of the North Carolina Botanical Garden or the Friends of Plant Conservation, please consider joining as a member. Membership is really critical to both of our organizations' missions. And thanks again for attending. We look forward to seeing you at other programs and other Lunchbox talks.